Hello everyone, welcome back to a new video. We're going to continue on our lecture here with the special senses. What I want to do real fast is to bring up that old drawing one more time. For those of you either didn't get to see it, got a slightly better internet connection today. So this is the one of the drawings that I'll be having you guys do in study group for those of you guys that come in for it. So, all right, so now what we're going to be doing is turn our attention to the eye today. And we're going to start off with the eye. We'll get into a little bit more in detail about it. But we are going to be discussing things starting off with the accessory structures of the eye. Now, with accessory structures, these are all things that are associated to the eyeball. They're not directly used in vision. They're usually protection, something like that. And the first things we want to talk about is the hairs, the eyebrows, and the eyelashes. And they're hairs to protect the eyes. Eyelashes specifically here on the eye itself, eyebrows above the brow line that can help protect, like keeping matter from getting into the eye, like sweat, for example, could be stopped by eyebrows, things like that, or other foreign objects getting into the eye. Then our eyelids, called the palpebrae, which there are two of them. There's an upper palpebrae, referred to as the superior palpebrae, and the bottom one, the inferior palpebrae. So superior and inferior palpebrae. The eyelids, and they cover and protect your eyes. So when you blink or close your eyes, these are protecting it from something that might be getting in there. Now, where the eyes meet, where the palpebrae meet, there will be the two canthuses, a medial canthus and a lateral canthus, found where the palpebrae meet each other is called the canthus. Now, also, there's a set of glands. Now, if we were to look, you would see the duct of them coming out in the eyelids. Now, this appears to be mascara from this individual and not the gland ducts, but perhaps the ducts maybe got a little mascara in them. But there will be a bunch of ducts opening up, and these glands are inside the eyelid, very kind of squiggly shaped ducts, and they are referred to as tarsal glands or meibomian glands. And they're very specialized sebaceous glands, and they produce lipids. And what they try to do is keep your eyelids, your palpebrae, from sticking together. Then, right here near the medial canthus, we will find the lacrimal carbuncle, which produces lipids when you sleep. If you guys look in your medial eye when you wake up in the morning, you will find that there will be some little pieces of material there. You might call that sleep in your eye. Well, that is secretions of lacrimal carbuncle. Now, another big thing, and what I wanted to do is I'm going to pause for 1 second and actually pull up my lab notes to show you a better view of the conjunctiva. So, like I said, I wanted to pull up the conjunctiva from here. The conjunctiva, there are two parts to it. The bulbar conjunctiva, which we're going to find located on the front of the eyeball here, and then it's going to fold over and form the palpebral conjunctiva. This is what we refer to, this is the area where you get pink eye, conjunctivitis. So these fold over as one flap, allowing the eyelid to glide over the eyeball. So the bulbar conjunctiva or ocular conjunctiva and the palpebral conjunctiva. And I'll probably just leave my lab notes in the background in case I need to refer to them again for a better image. Sometimes the things that were missing or something from my notes. Now, what we want to see is the muscles of the eye and what it is that they do. These are called extrinsic eye muscles. And what does it mean to be extrinsic? It's on the outside. Extrinsic eye muscles are external, and they control the eyeball's position, how the eyeball moves. Now, majority of these are actually controlled by cranial nerve number 3, oculomotor. Now, with the exception of the lateral rectus, and the superior oblique. Superior oblique, cranial nerve number 4, the trochlear, and lateral rectus is controlled by the abducens. Now, what we will see is these muscles move the eyeball around. Now, the superior rectus muscle, as we could see, elevates the eye. It would pull the eyeball up. The inferior rectus here would pull the eyeball down, looking downwards, depression. So superior rectus, you look up, elevates the eyeball. Inferior rectus looks down or depresses the eyeball. Now the superior oblique is a depressor. Now we got to think about this since it follows this weird thing here. When it pulls, it will pull and depress the eye and adduct. So that means it is down and in, down and in. Inferior oblique looks, it elevates and abducts. It's up and away. Up and away, the inferior oblique. Lateral rectus muscle, lateral rectus looks laterally, so we would say it abducts the eye, whereas the medial rectus pulls the eyeball medially, so that means it adducts the eye. Now, these muscles can become weakened and you get lazy eye. The nerves that control them can become damaged. Your eye will start to droop, will start to meander and wander. You get something called ocular ptosis, P-T-O-S-I-S, ptosis, something you'll see when, so you will be testing these in the cranial nerves when you guys learn to do neurological examination and discuss that. So this will be something you will be trained about in your future endeavors. So now beyond that, the other important thing is this area called the lacrimal apparatus. Now, what is lacrimal apparatus? Lacrimal apparatus is the thing that makes tears and transmits tears with the eye. So that means it includes the tear gland. The gland that makes tears is called the lacrimal gland. The lacrimal gland here, the lacrimal gland is a gland that secretes and produces tears. The lacrimal gland sends it through those tears, through these multiple lacrimal ducts, and the lacrimal ducts will conduct tears to the surface of the eyeball here. And then right here and here, there are two small holes associated nearby the carbuncle, referred to as the lacrimal puncta. And there are two of those puncta. A common punctuation mark is a period, well, a dot. These are two puncta dots. And they are connected to two lacrimal canaliculi. Canaliculi, canaliculi. So it's kind of like you're asking someone, can I lick your eye? Can I lick you lie? Not can I lick your eye, but can I lick you lie? Canaliculi. Little canals. There are two of these, a superior canaliculi and inferior canaliculi, that enters into the lacrimal sac. And the lacrimal sac, as it enters into the nasolacrimal canal of the lacrimal bone, the soft tissues become the nasolacrimal duct. Now, why does it get its name nasolacrimal? It is attached here to the nasal cavity and here to the lacrimal canal, running through that. So it goes from lacrimal canal down to the nasal cavity. And so this is why your nose runs when you cry, and you get a runny nose, because as you cry, those tears end up down into the nasal cavity. All right, so as we move forward, we're going to see the eyeball has three distinctive principal layers to it. It has what we call three tunics. The outer tunic is called the fibrous tunic. The fibrous tunic here, covering the outside of the eyeball is called the fibrous tunic. Underneath the fibrous tunic is the next tunic called the vascular tunic, which is the middle tunic or layer. Then the innermost here, more this kind of orangey color, would be the retina or the neural tunic. So from the outside in, we have the fibrous, vascular, and retina. FVR. FVR. Outside in. FVR. So the neural tunic is called that because it is the part that has the neural stuff in it. Now, as we go through this, let's look at our fibrous tunic. The fibrous tunic is the outer layer of the eyeball. It's collagen. That's why it's called fibrous. It's made of the fibrous protein collagen. Both the sclera and the cornea are made of it. Just the fibers are just oriented in different ways. Now, the white of the eye is called the sclera. The sclera, meaning it is very tough here. Sclera, the tough white outer fibrous layer. Then there is the clear area called the cornea. And just to warn you, eye puns don't get any cornea. So, ha ha ha. There will be a few eye puns that I will have to spring out in this lecture. Now, what happens is when the cornea here meets the sclera, there's what is called a or rather a cornea-scleral junction or corneal limbus. Corneoscleral junction or corneal junction, the limbus, something like that. We may call that, here it's labeled as corneoscleral junction, corneal limbus, corneal junction between the cornea and the sclera. Now, another thing, you could see it here, but I believe there's an image, I know I've got it in my notes in lab, is called the scleral venous sinus or canal of Schlemm. Scleral venous sinus or canal Schlemm. And right here, scleral venous sinus, sclerovenous sinus or canal Schlemm, we can see that best there. It is right here, that little hole that we can see. And if we zoom in a little bit, you can see that hole right here, right here. Now, what this guy does is there's a fluid being secreted right here, this little area here, the ciliary bodies, and they're producing this fluid called aqueous humor. And this is what delivers that aqueous humor back so it gets into the veins. And this actually reduces what we refer to as interocular pressure. And this is where something like glaucoma comes about. Now, one of the things that active ingredients in marijuana, the functioning drugs in marijuana that acts as a relaxer, helps to try to relieve that. It helps to get these aqueous humors back, reducing that risk. So now the joke I always make about this is, will the real Schlemm Shady please stand up? I know some of you will not get that joke because you don't know about Schlemm Shady. Anyway, that's always one of my worst jokes that I have to share. Now, what we will see next is, as we look at our vascular tunic, the middle layer, we're going to see that it's containing mostly the iris and the choroid. This is making most of it. Now, we're going to see other important structures and get into that in a minute. But the vascular tunic, the middle layer, has the iris. That's the pigmented part. What do we mean by pigmented? We mean it has colors to it. It is colored. It might be green or blue or brown, hazel, etc. And it has the ability to change the shape of the opening called the pupil. So it controls how much sunlight or light can get into the eye. Think of the iris as like the shutter of an old camera. It controls how much light exposure can happen to get to the retina. Now, the highly vascular part, when you see choroid or choreo, anything about choro, we are discussing vascular, like the choroid plexuses, chorionic villi, and the reproductive system, stuff like that. Now, you're talking about vascular. So also we will see the pupil. That's the opening in the iris, and light passes through it. We will see the lens. The lens focuses light. It takes the light, focuses it on what we call the photoreceptors found on the retina. Then we have our ciliary body, and this is the thing that we talked about right here with the choroid plexuses. Not choroid plexuses, sorry. The thing that makes aqueous humor associated to the canal Schlemm. I apologize. I'm getting other things in my head. I just said choroid plexus a minute ago. Now, the ciliary body is this area that makes what we'll see. That's one of the things it does is it produces the fluids called the aqueous humor. We'll talk about that in a minute, but it is mostly ciliary muscles whose job it is to change the lens's shape, focusing the lens, positions the lens, modifies the lens to focus on what we call the macula lutea, the retina. Now, I'm assuming that we've already seen this in lab, but I will take some time to show you guys these structures later on. Now, the ciliary muscles, they're the parts of it that fold to make ciliary processes, and they are called intrinsic eye muscles. They're inside the eye, and they control the lens and the iris. So these are important. These are the ones under autonomic control. They are the ones that, for example, if you have sympathetic innervation, your eyeballs your pupils dilate, they get big. When you have parasympathetic, they get smaller. That's controlling ciliary muscles to affect the diameter of pupil, the shape of the iris. So now one of the things we were talking about is this pupil here, this opening. There are these muscles. Now here you can see the ciliary body, which is made up of ciliary muscles that help control by contracting to make the lens get thinner or thicker, slightly changing its position, but mostly changing its thickness to bend the light that comes through it to make sure it focuses right here on what is called the macula lutea and the fovea centralis of the retina. Now, we will get into that more in detail in a moment. Now, vascular tunic, sorry, I hit my my headset. I've got a little bit of I'm getting over a head cold. So the vascular tunic has four major functions. And now let's think about this. There's blood vessels, lymphatic vessels. Those are vessels. So vascular tunic holds vessels. Wouldn't be surprising there. This is where your blood vessels and lymphatic vessels get into the eye. They provide a route for the blood vessels to go to eye's tissues for the venous drainage, the arterial supply, and lymphatic drainage of that organ, but also regulates how much light. Then it makes and reabsorbs aqueous humor involved in that, controlling interocular pressure because your ciliary processes, ciliary body makes aqueous humor, and also controls the shape of the lens for focusing. Now, we should know, based on the first chapter, that the ciliary muscles of the eye changes in diameter. We know that, but we should know that that is involved with autonomic nervous system. We should know that autonomic nervous system controls this, and this is going on. So we should know that's something that's happening. Now, what we're going to see is that parasympathetic nervous system and sympathetic nervous system control that as well. Now, one of the things that controls the diameter of the pupils that is very important, iris's shape, diameter of pupil, is going to be the amount of light. And let's talk about this a little bit. So let's say right here that I've got two eyeballs here. So here's my eye. I need to make that a little bit bigger because reasons. So let's get that on there a little larger for us. I'll need two of these. And in our eyeball here, we're going to be looking at that. Here we have a pupil. And let's put both pupils at a normal diameter. Oops, my air server just lost connection. Probably because, yes, it went back down to a very low connection. as other people are here. It's always very frustrating to try to do anything with this. My office is in a very poor location to connect to the internet here, Wi-Fi for my iPad. Let me pause and see if I can fix it. Okay, we're back. So let's say I've got two eyes here. And what we're going to do is on this eye, we're going to keep this one here normal. Now, to control this, we have two muscles. And I'm going to draw rings around this here. And then let's use blue. And let's start that out normal. Let's see if this will catch up to me. Now, what we have are radial and constrictors. Let's see how long it takes for that to play catch up. Okay. So there is a circular constrictor muscle here. I'm going to see if this thing actually will work. So the constrictors are circular. And what we do is, so I'm going to need to do two separate drawings here, is let's say I've picked up a flashlight, and I take this flashlight here, and I shine this light into your eye. Now it's going to take a minute for this to actually work because this thing is very slow. Really struggling to try to do what it's supposed to do. So I'm going to see how long this takes. I may go ahead and pause and wait for it to come back. Okay, so we shine a light in the eye, and there's too much light. So if there is too much light, we want to make sure this pupil gets a little smaller. So how are we going to make that pupil get smaller there? How do we do that? Well, what we would do is we are going to need to get our constrictor muscles, and we're going to need to really squeeze them. So as they squeeze and they will squeeze in, they will push inward and they will make the diameter much smaller. So they're going to bulk up. They're going to squeeze and get thick. And your radial muscles will stay relaxed. So that will squeeze down, making that pupil a little bit, quite a bit smaller. So kind of choking it down, so to speak. So as it does that, that pupil diameter is very small. Now, if we were to take the same thing here and then take a normal eye here and have no light, and we make it very dark for this person, more light needs to get to the eye. Not enough light's getting in. So that means now your pupillary constrictor muscles, they will relax. But what will happen is these radial dilators, they will beef up. They will contract and shorten. And as they get beefier, they will pull this open. And what they're going to do is open up that pupil and make that pupil get quite a bit larger, letting in more light. So it will open up because the radial dilators here, the radial muscles, they will contract and shorten and pull that iris open more, letting more light in to be able to see. So it's trying to accommodate a negative feedback for decreased light. And then here, it is a negative feedback for too much light. Okay. And I'm going to go ahead and move to a blank page here. There we go. So I just didn't want it to freeze up. All right. So I hope that made sense is what I was drawing out there. Our book doesn't do it, so I wanted to draw that out. So too much light enters the eye, the diameter pupil must go up. It's because the constrictor muscles contract and they make the pupils decrease, get smaller in diameter, contracting to decrease it. Now, when there's too little light, the diameter needs to go up. The pupillary dilators, they shorten, they pull the eyeball open. Now, parasympathetic constricts the eyeballs, making the pupils smaller, rather, and sympathetic makes the iris get bigger. It increases the diameter of the pupils. So they are opposites. Remember, parasympathetic and sympathetic are always opposite in all ways, in terms of actions, in terms of anatomical things, pre and post ganglionic length, etc. So here you could kind of see this as well, the dilators and the sphincters. I call them, they have them here, sphincter pupillae and dilator pupillae, but this is kind of how I drew it. It's a little bit different, a little bit more simplistic, but you could see that the two things affect the eye to get bigger. Now, for example, if somebody had a really huge dilated iris here, that could be something done while you go get your eyes dilated during eye exam with atropine. Stuff like that. And that would mean that it's going to stimulate the sympathetic stuff. And actually, back in the day in Victorian England, when people would go off to see the opera, they would put a tincture of belladonna in their eye to dilate their pupils to be able to see better in the dark opera house. It's kind of cool. All right. So this is going to be the first really difficult concept, and I would say the most difficult concept for the eye is how light refracts. Refraction of the eye. And I will be hitting this as one of those deep concepts. Now, light is refracted. What do we mean by light is refracted? It gets bent. Anytime light passes through anything that it can pass through, a medium that allows light to transmit through it, it is bent. Now, what does that mean? If you've ever seen, for example, over top of a hot grill, when you're grilling, like on the 4th of July, and you're grilling those hamburgers and hot dogs, and you look above the grill and you see the heat waves above that, or above hot asphalt, or in the a hot parking lot. When you see those waves of heat, that is changing the density of air, making light bend through it differently. And that's how you get like mirages. It looks like water when you see it. Looks like standing water on the sands in the hot desert. But really, it's just the air is now hotter, so it refracts differently than cooler air does. And water, it refracts light different. Air reflects light different. A lens, like a crystal lens, it refracts light differently. So every time you pass through a medium, it bends it, but it bends it differently. Now, we are going to say that an image, if it's focused, only when it gets to the most sensitive part of the retina. Now, let's explain some stuff here, okay? Let me help you guys understand some optics. Now, let's say here that you were, that we have a lens, and on this lens here, we got a lens, and this lens, any lens that exists has a point in which a lens focuses. So let's say I've got an individual here that we're checking out, that we're seeing that they're a cute, attractive individual, and light for them comes into our lens. That light passes through and is refracted and is then bent by that lens onto an area called a focal point. And I'm going to abbreviate it FP. Now I'm going to pause my recording and let this catch up. Okay, so it caught up to me here. So we have a focal point, and that focal point is based on that lens. The distance that individual we're checking out is away from us, and a few other physical properties. So now, let's say I drew a line here right down the middle of this lens, and I'm going to draw it as a dotted line, and we drew another dotted line here at the focal point, and I were to measure the distance between my lens and my focal point, we would call that a focal distance. Now I'm going to pause and let that also catch up. Oh, I thought it did there. It's just trying to get focal distance. It's very slow, as you can tell. There we are. Okay, so very, very slow this process can be sometimes. Okay, so we have these major things here. So if you're drawing this with me, hold here, have this a drawing that looks like this. I do have a YouTube video on this as well, and I'll make sure to let you guys know about that. So what I want to do is discuss that there are focal points, and the focal point is wherever it focuses. Now, that's just, all lenses have focal points. It depends on the lens's thickness, distance of the object away from the lens. But if it is focused, then it's clear. If it's not focused, it's fuzzy. And we want to make sure the focal point ends up on the most sensitive part of the retina. So for the eye, the lens's job is to refract the light onto a focal point where it is focused onto the retina. That's what it should do. Will it always do it? No. Now, the distance between the lens and the focal point is called the focal distance. Now, to affect focal distance, we have two major things. The distance an object is from the lens and the shape of the lens. Now, the objects, let's talk about that. And I like to just do it this way. So, this individual here in black, they have a certain focal point right here in black. Now, let's say this person saw me checking them out, and they thought maybe he looks cute from a distance. Maybe he's cute. And they walk close towards me to come and check me out a little bit better because they were moving towards me. I'm going to put a smile on their face because they're kind of looking at me, and maybe they think I'm cute. And if they think I'm cute, they might want to move towards me. So they got a smile on their face. They move towards me. Now, this means that the new focal point must move with the individual. So now their new focal point's over here. So here we have a new focal point based on the lens and where it is. Now, let's say right here, and what I'm going to do is I'm going to pick purple. And I am going to come right here and say that that is my retina, and this is where I want vision to be. Let me pause and let this catch up. And let's say this focal point in black is the fovea. So let's just say that is the fovea, okay? And this is the ideal location, the black focal point. Okay, so this is our ideal fovea location. And now what we want to do is imagine that that person's distance that they move towards us has now changed the focal point. It's on the fovea anymore, so now it's blurry. Now let's say that this person here as well, that they noticed that they got up close, saw I was not cute at all, decided don't want to check this guy out anymore. I'm walking away from this. I'm moving away. He is not a good looking guy. Man, we're going to put a frown on their face. We'll make them red, make them move away here. So let me pause and let this catch up. Okay, so what we could see is they moved away from me. So this means now that the old focal point is now got to move too. And let's say it's about here is the new focal point. Now that means that focal point is also blurry. So let me pause. So here we can see that new focal point. So what you're going to see, if I were to go here, I would see this is blurry. And I would see this is blurry. And in order to make, so we see that the distance an object is from the lens affects the focal distance. Let's look at that now. Okay, so let me help you guys understand this. First, let me actually go back and find the green that I was using. Okay, so what we saw when an object moved near the lens, we saw that that focal distance was now increased. We saw an increased focal distance. So we saw an increased focal distance when the object moved towards us. And then when the object moved away from us, we saw a new focal distance here. It decreased that focal distance now, and we saw a decreased focal distance. Okay. So objects, when they move near to lenses, increase the focal distance. When objects move away from lenses, they decrease the focal distance. Now, the other thing, so that's the first factor that influences focal distance, is the distance objects are from the lens. The other factor is going to be the thickness of the lens or shape of the lens. Now, the thing with you that I want to make sure you guys get an idea of is the rounder the lens, the more it bends. The rounder the lens, the more it bends. What does it mean to bend? Refract. The rounder a lens is, the thicker a lens becomes, the more refraction it does. So that means a round lens, a thick lens, bends light more. So that means it reduces the focal distance. Now let's think about this. When would you need to shorten the focal distance? Well, look here. In green, my object has moved so close that the focal distance is too far and now blurry. If I could shorten it somehow, I would be able to see clearly. So what do? The rounder the lens, the more it bends. And that is an easy way to remember this. So what we're going to do is we're going to make this lens get thicker. We're going to take the muscles and push the lens in and thicken that lens. And what we're going to do is we're going to take this light from here and we're going to bend it back onto the fovea. And it will be clear. Now I'm going to see if this, I think it's going to take a little while, so let me pause. Okay, so we now have put it on the fovea where it will be clear. So now we've seen. Now, as we've seen over here, let's step back to the other thing, is that when we decrease the focal distance, when the object moved away, the red individual, the red bit, when they moved away, we now have a focal distance that's too short. We're undershooting the focal point, the fovea, and we need to make it clear. How are we going to make it clear? Well, if the rounder the lens and the more it bends, well, here's the deal. If we make, when it's red, if we make it too round, we're going to make it even worse and never make it worse. So what do? We need to make the lens get thinner. So we need to flatten the lens. So now what we're going to do is make the lens get more flat, and that's going to be the first thing. Finally, that took forever. So the lens gets thinner, and as that lens gets thinner, what we're going to see is we're now going to bend the light less. Because if the rounder the lens, the more it bends, we're undershooting, so now we bend it less, and we take this focal point and we move it to the fovea and make it clear. I don't know how long this is going to take, so let me pause and give it time to catch up. Okay, so there we are. So if you can draw this out, and I recommend draw it repeatedly to be able to get that down, because this will be one of the topics that I have some of the more difficult questions on for my exams. Okay, this is one of those kinds of things that if you understand it, kind of the higher order thinking levels of questions that I do. So even if you're in my internet course or my standard course, because even my on ground people may be watching this on YouTube. So all right, so screenshot this if you want, but you need to draw it to get that out of it, because this is going to be a really helpful drawing to do if you take your time. But also, I have this done on YouTube. Sorry, let's click on the wrong thing there. So if you follow this, if you go to my YouTube channel, I have a whole playlist on senses where I have a lot of the stuff we're going to do and some extra stuff that we don't do in this class. For example, vestibulo-ocular reflex, things like that. All right, so is the sound not working? There we go. I got nervous there. Okay, so if we understand that, then we can definitely do the most difficult things. And here's a few little pieces showing that, but I wanted to go through that with you guys and draw it out. Now, refraction, bending of light, occurs in 4 major places. Your cornea bends light. Now, if it's got an unusual shape, if it's rough and bumpy, for example, what happens in astigmatism, that can cause refraction to be messed up. The aqueous humor, so if you have too much of it, aqueous humor can definitely mess it up. Your lens thickness, for example, lens is the major refraction area. It could be too thick or too thin and have problems causing nearsightedness, farsightedness. And the vitreous humor, the fluid that's in the back of the eyeball in the posterior cavity of the eye, something you would talk about in lab. All right. Now, so what we basically just learned that we've drawn out together is accommodation. Accommodation is where the body automatically adjusts the lens's thickness to give us clear images even if something is distant or close. When something moves in close, remember a close object has a greater focal distance. So the focal distance might be over here, but if the lens thickens, it pulls this back, boop, here to the fovea centralis and makes it clear. When something's far off, the focal distance is too short. Lens gets thinner and flatter to be able to increase the focal distance to put that on the fovea centralis to make it clear. So when objects are near, the muscles, the ciliary muscles here, they contract, as you can see, they are contracting, and they will push and thicken the lens, making it more round and bending the light. When things are far off, the ciliary muscles here relax, making the lens thin up, going more flat, thinner, bending the light less. Now, you take this and what we just drew here and combine that, and I'm going to ask you for when objects move towards or away, what happens? How does that affect focal distance? How does that affect lens thickness? How does that affect the ciliary muscles? Okay, and I'm going to ask you questions associated with that. I may ask you, will all of these happen except for what? And there'll be everything does happen except for one thing, A, B, C, D. Now, my exams are all multiple choice, as you know, but anyway. So I want to make sure you knew that. Now, there is our normal vision. Normal vision is called emmetropia. Emmetropia. Now, normally, most of your life, you're looking ahead of you. I know sometimes we feel like we spend most of our time looking at our past, looking behind us, or looking at our feet. But really, most of the time, a human being is looking ahead to see what's coming at them. That's just a thing that animals like us do. We look off into the distance, looking to see what is coming at us. And to do this, we want our ciliary muscles to be primarily relaxed, relatively relaxed, and have a thin, flat lens, because we're trying to see what comes off at our distance. And that means things far off are going to be more in focus. We're usually looking ahead, what's coming at us before it gets to us, not what's right in front of our face. Sorry, I've been getting tongue-tied today. Now, we usually measure visual acuity at 20/20, saying that you see at 20 feet what normal people see at 20 feet. And let's say, though, you have 20, like if you have 20 10 vision, something like that. If your vision's 2010, you see at 10 feet what you see at 20 feet, what most people have to be 10 feet away to see. 20/30 vision, you see at 30 feet what most people could see at, or you see at 20 feet what most people can see from 30 feet away. So you have to be 20 feet away. Sorry about that. Sometimes I mess that up a little bit. Okay. Anyway, that's not something we're going to be testing on. Now, myopia is nearsightedness. One of the things I will ask you guys is what is the cause of either of myopia or of hyperopia? Now, myopia, that's nearsightedness. Now, what does it mean to be nearsighted? Why do we call it nearsighted? Well, there's two probable causes, two common causes. Eyeball is either too deep. That means that when the focal distance is measured, it is too short. Focal distance is too short, too great of a focal distance. Or the lens at rest is too thick. If it's too thick, it overbends the light. Or the eyeball is too deep and the light does not hit the fovea. So that means anything far off is going to be blurry. You're not going to see it. And you can only see things nearby you, hence nearsightedness. So the two common causes is either an eyeball that is just as it developed was too deep, like here, or a resting lens thickness that is too thick. You will see things far off are blurry, and only things nearby you are clear because the lens may be too round. Now, to fix this, we use a lens called a diverging lens. And what this lens will do that we put in front of it is that it will spread the light out. And when it hits our lens that either bends light too much, or an eyeball that's too thick, it will fix and correct and increase the focal distance for us. It spreads the light out a little bit, so if we have a lens that bends light too much, it compensates for that by spreading it out, diverting the light, and so that our too thick of a lens does its thing and accounts for that, accommodates for it. Or the eyeball's too deep. Either way, you're going to increase the focal distance and make it visible. So one of the things I always say is think of Dr. Myopia, MD. Dr. Myopia, MD. Myopia needs a diverging lens, MD. Myopia needs diverging lens. And I will test on that. Now, hyperopia or farsightedness. So I'm going to test you guys on hyperopia or myopia. What is the causes and what kind of lenses would you want to put in? Now, one of the things we would see is either the lens is too thin, resting too thin, so it doesn't bend the light enough, and that causes the focal distance is too long, or the eyeball is too shallow, again, making the focal distance too long. It is causing an increased focal distance. So anything that is close to you will be blurry, but you only see things far off very easily. This is called hyperopia or farsightedness. Now this is, think of high C. Remember the drink high C? High C. Hyperopia needs converging lens. High C. Hyperopia, converging lens. The converging lens is a thick lens that bends the light extra, that is going to bend the light more for the too thin of a lens that you have or the too shallow eyeball, and it accounts for that the focal distance was too long, and this lens shortens that focal distance for me, making it seem normal. Whether this lens is a contact lens or whether it is a glasses lens. All right. Now, image formation. I want to talk about this very briefly. It's not something we're going to spend a huge amount of time on, but objects, light from the top of an object goes to the bottom of a retina. Light from the bottom of an object goes to the top of the retina. Light from the left of an object goes to the right of the retina. Light from the right of an object goes to the left of the retina. So everything that goes to your retina is an inverted mirror image. It's upside down and mirror flipped of everything you see on the retina. So your brain is able to go in and say, hey, this isn't right, and flips it. But sometimes people may get a head injury and they may not be able, they may see the world as upside down and other way around. So the wrong mirror, inverted mirror image. They may see the world upside down and mirror imaged. But our brain knows this, it corrects for it. So I do talk about that. Now, as you can see that here we have some objects. Same thing here, left from right, flipping, and top from bottom flipping. So everything is inverted and mirror imaged. Okay. Now, given that, now what we want to do is turn our attention to the retina tunic, the neural tunic or retina, and that's the inner tunic. So let's sketch this out real quick. Since you've seen this in lab already, let's do a sketch. And what we're going to do with retina is, so let's on top of here, let's just go ahead so we know this is retina, is right retina. Now, what we want to do is at the very top is draw a block of cells, and I'm just going to make all these little boxy cuboid epithelial looking things, and I'm just going to put a dot in their center representing nuclei, but I'm drawing it in black, and this will be the pigmented epithelium. Now, just because of space, I'm going to abbreviate things. The iPad's taking forever to do this, so let me pause and let it catch up. Okay, so we have the pigmented epithelium. Now, this is the first kind of layer of the retina. This is, as we move just behind here, you're going to have vascular tunic. Just so you know, it's right behind there. And then this is the inner surface of retina. Inner surface of retina. Okay. Now, after we hit the pigmented epithelium, we then enter an area here called the neural portion. And I'm going to abbreviate that NP because just of space and everything, the tablet I'm using to do that. So, the neural portion. Now, all the way in the back, you are going to have some cells called rods and cones. Now, rods will be quite numerous, so I'm going to draw some rods. 1, 2, 3, 4 of them. Let's draw 4 rods. And I'm just going to put R in them for rods, and we're going to draw these guys with black and white. So these will be our rods, and I'll draw 4 rods. Let me pause my recording. Well, it's been a little while. If you could actually see the clock in the bottom, you can see how many minutes have gone by, and it's still not done anything. So I am just going to move on and hopefully be able to draw some more in a minute. So what we were seeing is there was a pigmented epithelium. This layer is kind of dark. It has a lot of the pigment melanin in it, and that melanin is going to absorb any of the excess light that's absorbed so that it doesn't reflect and you don't doubly stimulate your photoreceptors. Now, the 3 principal cells that are present in the neural portion, there's 3 layers of the neural portion. The photoreceptor layer, the bipolar cell layer, and the ganglion cell layer. Now, the photoreceptors are your rods and your cones. That's what's found in there. Then your bipolar cell layer, that's going to be your layer of cells that synapse with the photoreceptors and the ganglion cells. And then the ganglion cell layer, and that's going to be synapsing with bipolar cells to form the cranial nerve number 2, the optic nerve. Let's see if this has at all done anything. No. So what I'm going to do is in my drawing, I'm going to go ahead and just draw it. And just be able to do this. And I'm just going to draw it, and hopefully it will catch up. So that would be my hope. So let's get some cone cells in here. Let's get some bipolar cells in here. And then I'm going to get a ganglion cell. Let me use that. I like to use green for this one. G, ganglion. And I'm just going to draw it out, and hopefully it will eventually show up. And that will be the goal here. And then that way, when it does work, you'll be able to see it. And it's taken me forever to do this because of that. Um, as that goes through, I'm going to let that work in the background, see if it connects, and I'm going to go through the rest of that. So rods and cones. Now rods, remember, rods are in dim light. They're most numerous. There's a lot of them. There's many rowdy rods. You got a rowdy party because there's a lot of people there. The rowdy rods, there's a lot of them. And they are doing blacks and whites. They see the world in black and white. They are working in the dark. So since they are in the dark, rod, dark, and cones see color. C for cone, C for color. Now, what they do is they are sensitive to, there are what we actually call red cones, green cones, and blue cones. And they're different sensitivities of different wavelengths of light. Now, wavelengths of light, for example, there's different wavelengths in nanometers, ranging from around 400 nanometers to 700 nanometers in wavelength. Do not worry about those numbers. That is just the visible spectra of light, where the different, like blue cones absorb mostly the blue light, and you can see their absorption here is mostly absorbing in the blue area, the height of the peak, and the green cones and red cones. And rods do darks, so they do less of the bright, and they absorb mostly in the dark. And you can kind of see that they do different lights. There's red cones, green cones, blue cones, the three primary colors. And there's different wavelengths that they absorb to see colors. Now, because of this, they're less sensitive. They need more light to activate them. So if you think about this, if you're in a dark room early in the day and you're trying to find your socks, so you get socks that match your pants, if it's dark, you may not be able to see what color those socks are very well and get socks that are on color. So what you need to do is have bright light. You need to turn the light on so you can see what color clothes you're getting out. Now, your bipolar cells, these synapse with your photoreceptors and ganglion cells. Now, bipolar cells can synapse, you can have several rods, so more than one rod to a bipolar cell, but only one cone for a bipolar cell. Okay, this is a unique thing. Then the ganglion cells receive information from bipolar cells, synapsing with those, and they have an axon that forms cranial nerve number 2, the optic nerve. Now, if it's working, you can see it started to try to catch up to me in the time that I've been talking, and this is all that it's done. So let me pause and then let that catch up. Okay, so it caught up with me. Here we go. So, this is a drawing I do in my lab as well. All right. So as we move along, so we said that C for cone, C for color, that the cones do different colors of light, different wavelengths. This is something I want to talk about. Now, the ganglion cells that synapse with your bipolar cells, there are two types of ganglion cells. The M cells monitor rods. M cells monitor rods. Now, they're big, massive M cells. They're massive. They do general form of the object, motion, shadows, and dim lighting. P cells, the other one, does the cones. Now, they're smaller types of ganglion cells. They are referred to, they're the petite And there's a lot of them, so they're plenty. Petite and plenty, P cells. They monitor cones, and they tell me about like the edges, fine detail, and color. Now, it's these cells, the M cells and P cells, that camouflage interrupts, and that's how it works. That's what its job is to do, is to mess up your M cells, and that's what camouflage is doing. So that's relatively interesting. Now, you could see it here. We could see the inside of the retina. We could see the ganglion cells, the bipolar cells, the rods and cones, and the pigmented epithelium. This is what we drew here. So if we put those side by side, we can put them kind of side by side there. Sorry, I actually moved my iPad over to another place to let it kind of charge, because I won't be drawing for a little bit longer. So, all right. So if you are looking for a good time to take a break and pause, definitely would be it. But I'm going to move on and look at some of the rods and cones. And this is also the second big thing for today, the second difficult thing. So the first really hard, difficult thing is to understand this refraction all in one slide, and this is the next big one. I do test on it. They used to do it in departmental final, but they stopped doing it. But I do test on it myself. It'll help you understand diseases like night blindness if you get into that, and pathophys. So there's some things here I do want to discuss, and I will test on it. Since I'm going to teach it, I might as well test you on it. So now the rods and the cones, they have two different segments. An outer segment that contains the visual pigments that we're going to talk about and optic discs found here. And then there's the inner segment, and that contains the area where neurotransmitters get released from, okay? Now, the other things, there is a stalk that will be associated to those. There are stalks on them that attach the inner segments to the other part. So there is a stalk. There is a cell body in them. So there's connecting stalks. You can see the connecting stalk here. You can see the cell bodies holding the nuclei, and synaptic terminals as well. And they contain a neurotransmitter found in vesicles called glutamate. Glutamate is the neurotransmitter of a rod and a cone. Now, we are going to be talking mostly about rods from here on out, because the physiology of a rod is the same as the physiology of a cone for the visual function. It's just the different pigments and different wavelengths that they absorb is the difference. Okay, so what we want to see is there is on a rod, there is this area here that has the discs in it. These discs are studded with these proteins called rhodopsin. What rhodopsin is there's an opsin protein, and inside a retinol molecule. So this is where you get rhodopsin. Rhodopsin is retinol and opsin protein combined together. Now, on this, I normally try to draw this, but my iPad is just not going to work, so I'm going to use the diagrams here. Now, what happens in visual physiology is I'm going to preface this with that when it is in the dark, when it is darkness, when you are in the dark or you're asleep at night, your photoreceptors are releasing glutamate and letting me know that a bipolar cell that it has not absorbed a photon. Why? You are actually awake less. You're asleep during different parts of the day, and you're awake. So what this means is, since you are actually asleep less than you're awake, we can save glutamate this way. So you're actually releasing glutamate when you're not seeing, and you don't release glutamate when you are seeing. And this is because you're awake more time in the day than you are asleep most of the time. Now, to do this, we need to produce a dark current so that I can release glutamate during the dark. So how do we do it? Well, let's imagine you're sitting at home, you're sitting here at the house, you're bent over that's a retinol molecule. You're bent over your notes. You can see it's bent here. You're bent over your notes and you're trying to study your notes, and you're at home. It's a lovely day. So what you've done is you put a door stopper in the door to let a lovely breeze come in, and this breeze comes in and flows out. So it comes in the outer segment and leaves the inner segment. Sodium flowing in is the breeze. Okay. We have a doorstop, and that lovely breeze is blowing in, but it's going to make it difficult for you to see your notes. It's blowing on you, moving your notes. You can't see it. So what you're going to do is you're going to tell somebody, your family members, to go and remove the doorstop so the door is shut and you can see your notes. So how does that happen? Well, that's what we're going to talk about. But dark current is when you've got your door open, you have what is called a gated sodium channel, and it has a door stopper in it called CGMP, which stands for cyclic guanosine monophosphate. So what is that? It's a guanine, remember GTCA of DNA, bound to a mono 1-phosphate. So basically, it is like ATP, ADP, AMP, without adenosine. It's just got guanine. So it's guanosine monophosphate. And it's been cyclic structure. So that's why it's a ball. Now, what's going to happen is when that's there, sodium comes in the outer segment and flows out the inner segment of a rod or a cone. As it flows, this is called dark current. It keeps it about negative 40 millivolts, so you're actually quite low, quite negative, and it releases glutamate. So it comes in the outer segment, sodium, and flows out the inner segment. Okay, so this is called dark current. Now, remember, you were retinol sitting here. You're retinol. You're sitting here bent over your notes trying to see, but you can't because the breeze is coming in too strong, dark current, and you want to turn it off. So how do you do it? Well, you are retinol. Now, retinol comes in two conformations, 11 cis and 11 trans. Now, once upon a time, my sis gave me at Christmas a Crown Royal bag full of $1 bills. Now, I thought maybe there was something wrong. Now, I knew what a Crown Royal bag was. I knew Crown Royal was rum. It was a liquor. And I don't want to know how my sis got all those $1 bills. But I bet my sis was a little less than straight at that point. She probably was not very linear. She probably was a little crooked. She was my 11 sis. Because I don't want to know how she got all those $1 bills, but I'm not asking. But basically, it was a Crown Royal bag full of $1 bills I got for Christmas from my sister. That tells you some things about my family, and I'm the one who's not allowed at Christmas dinner. Anyway, so what happened was when the photon hits it, a packet of light, strikes this 11 cis, it converts it to 11 trans. It makes it straighten out. So now my sister is she's living at the foot of the cross now, and she straightened herself out because she saw the light. She saw the light, and she straightened herself out. She's been transformed. It's a straight and narrow from here on out, and she's 11 trans. Now, when that happens, when that photon straightens it out, it's kind of like you're sitting there and you stick out your arm and point your finger at one of your family members whose name is transducing, and you say, "You go shut the door." Well, it doesn't shut the door. What does it do? Instead of going to shut the door, it tells PDE, phosphodiesterase, PDE, to go break down cGMP. So what is it going to do? So transducin, retinol, tell it to go break it down. Well, retinol doesn't really do it. Opsin is activating it. That is true. And as that happens, it goes and activates PDE to break down cGMP. When that happens, you're basically going back here, and cGMP is like a doorstop, and PDE, which is right here, comes in, knocks that out, and the door shuts. And that will diminish dark current. Okay? So dark current diminishes and glutamate stops being released, and the bipolar cell knows that we've done this. So what happens is, step one, photon hits this 11 cis retinol, turns it to 11 trans retinol. It straightens out. 11 cis means it is not absorbed light. 11 trans means it has. Now, actually, what will happen is when that happens, this molecule detaches and it exposes an enzyme complex that activates, this enzyme complex activates transducin. It actually has to do it 2 times to make transducin here activate. Transducin activates PDE. PDE comes in and breaks down cGMP, closing the sodium gates, and that actually acts like a doorstop now, and the door shuts, and dark current diminishes. That means no more sodium flow. That means glutamate release stops. And now the cell goes to normal resting potential, about -70. It hyperpolarizes to C. Now, wait a minute. Why would you want to hyperpolarize it to make it C? To go from -40, which is more positive than -70, why would you want to do that? Why would you want to go from -40 down to -70? Why would you want to hyperpolarize it? Well, one of the main reasons, we've already said that because you are asleep less during the day than you are awake, this saves glutamate. Okay? But here's the other thing. What if you had to form an action potential every single time you wanted to see? You would be blind during the absolute refractory period. And that wouldn't be good. You would see blurs in your vision would be choppy. It would be flashy. You wouldn't see it as one smooth image. This is why we do it this way, so that you would not be blind during the absolute refractory period of a photoreceptor. So what we just do is we just turn it off. The default position is on, and we turn it off when we see something like developing a film, and then we go back to its original position. So it's telling it whether it's been turned on or turned off. It's telling it that a photon had actually turned it on or off. If it absorbs a photon, we turn it off. Okay. So what we do is our bipolar cell is receiving information from our rod or cone, and that sends it to a ganglion cell to be known about. Now, photon strikes, 11 cis, my crooked cis. She saw the light. She straightened herself out. Then that detaches, and when that detaches, that actually will activate transducin to activate PDE to break down cGMP. Then you actually have to put this back together again and detach it. Now, that is actually right here is why when you go out in the bright light and come back into a dark house, you can't see jack squat, is you're having to reassemble that so you could see again. So this reassembling here, we call this rhodopsin bleaching. Retinol and opsin is bleached and has to be reassembled to see again. That's why everything looks so dark when you come in from the bright outside to a dark house. That's why you can't see anything. All right, so that is our next really bad one. If you think about this kind of like my analogy here with the doorstop you're trying to tell your family, hey, you go close the door. You tell transducin to close the door. Transducin's like, I don't want to go do it. Tell PDE to do it. PDE breaks down CGMP. That means it just kicks, it doesn't shut the door, it just kicks the door stop. The door shuts. The dark current stops. The wind stops blowing in. The sodium stops flowing in. The cell hyperpolarizes, and bam, there we go. All right. And I've actually been asked that many times by people out there on my educational page and things like that. Why is it done that way? Well, that's why. It's just ultimately why. Now, at least the best answer I could give you. Besides that, I said, however you think the human body got here, either evolution or God did it. So that's the answers. But I'm going to say that's the good reason to do it. It saves glutamate, and you won't be blind during the absolute refractory period. Okay, now in the retina, we also have an ora serrata, which is where the retina meets part of the vascular tunic. It's the ridged or serrated anterior edge of the retina. There is a macula lutea. Think about the macula lutea is like a target, and that's where cones start to concentrate. But at the center of a target, there's a bullseye. The bullseye is the fovea centralis, and that is where there are only cones, no rods, only cones, and the sharpest vision is there. So if a lens can refract light onto fovea centralis, it is that you have the best vision. Now, the optic nerve is made by the axon of ganglion cells as it leaves the back of the retina, and where it leaves is called the optic disc. And because of this, there is no photoreceptors there, so it's a blind spot. And everybody has one. If you ever do a blind spot test, where usually you will see like a star and a square or a plus sign, some shapes that you look at, you move it towards your eye when it disappears and measure that. That helps you figure out the position of your blind spot, things like that. But we all have one. Even the best vision has them. It's just that can be there's variations of blind spots. Now, in front of the eye here, we have the lens. Let's take the lens as a landmark. In front of the lens, we have what is called the anterior cavity. Behind the lens, we have posterior cavity. Now, the anterior and posterior cavities hold different fluids or humors. The anterior cavity holds aqueous humor in front of the lens, everything in front of the lens. Behind the lens, the posterior cavity holds vitreous humor. Now, anterior cavity is between your cornea and your lens, holding aqueous humor. Now, if we take this lens in front, then we take our iris here. In front of the iris, we have the anterior chamber of anterior cavity. And because the pupil is an opening to this anterior cavity, there is a posterior chamber of the anterior cavity. Okay. So between the iris and the lens is your posterior chamber of anterior cavity. Between the cornea and the iris is the anterior chamber of the anterior cavity. This is a cavity that's so big it has two chambers in it. Think of it that way. Think about a big, nasty cavity in your teeth that's so big it has separate chambers. Even though the anterior cavity is the smaller of the two cavities, the larger cavity being posterior cavity, full of a gelatinous vitreous humor. All right, so our very last thing that is really big is going to be our visual pathway. I will test on the visual pathway. I'm going to ask you about which fibers cross over, which ones don't. I'm going to suggest you draw this with me. So let me pause and come back, and we're going to do another drawing. All right, so here's your last chance to get this drawing here. So pause if you want to get it, or rewind back to it. All right, now I'm going to move to a new piece of paper here. It's going to take a minute for this to show up. What we're going to be talking about is our visual pathway. Now, to draw the visual pathway, what I want to start off with is if we were to draw two cups here and here, two little cups, basically. It hopefully will come up in a minute. We're going to draw two little cups. Let me pause and let it catch up. Okay, so there's our two cups. Then we're going to start here and we're going to go like this. Let me pause and let it catch up. We're going to do that. Then we're going to come back like this. And draw something that looks a little like that. Sorry I didn't get it as nice. I can do it a lot better on a board. So now what we want to do, let's label some things. We have our retina. We have our optic nerve. We have our optic chiasm. We have our optic tract. And then we have our, remember, if you want to hear me, medial geniculate nucleus. That was for the auditory pathway. Here we have lateral geniculate nucleus. Geniculate nucleus. So if you want to look, lateral geniculate nucleus. Okay, now what we're going to do, let's get two colors. Let's do Christmas colors. Here I'm going to put medial, visual, field. So I was going to say medial vis field. And in green, let's do lateral, visual, vis field on both sides. Lateral, vis field. Okay, let me pause and let it catch up. It's trying to. Let me pause again. All right, it caught up. You can see how long it took. Okay, so what we then want to do is remind ourselves that light from the right of something hits the left side of the eye. So medial visual field information here is going to strike the retina that way and the retina this way. Okay? And then lateral visual field information is going to strike the medial retina. Light from those areas. Let me pause and let that catch up. All right, I'm not going to wait anymore. This thing is just too slow now. It's I am going to use this. I like to do it the way that I draw it. What I went ahead and did is pulled this up. If you follow on my YouTube channel, Myelination, I do have where I actually go through and draw this out. So you could see. And you could see me go through that, and then we label the structures. Then we get the visual fields. Now here's the tricky part. So this is what you want to practice drawing. Now, what I do is I also go in and make some other things I talk about. Then I do make some lesions. These are just a little bit more advanced topics. But if you can do the pathway, that is the important part, and there I do have a video drawing that out. I do apologize that the drawings didn't work so well. So what we have is medial visual field information here. It strikes the lateral side. And then here, again, lateral sides, where lateral visual field information strikes the medial sides of the retina. And then that information does cross, is only lateral visual field information crosses over. Lateral visual field information crosses over. Medial visual field information does not. So the way that I do mine is this drawing here. I believe is a little bit easier way of seeing it than what our book does, and a little bit more simplified than what our book does. But actually, if you know this, you will talk about this in nursing classes for sure. I know in other places, in other healthcare fields, they will discuss it. It is something you will discuss there. Also, some people have done the visual pathway in lesions, like Armando Hasugadom. I do really like his stuff. I'm not just trying to say, oh, go for Mr. Lefford's channel. It's just I do produce some videos. His is a lot better quality because he's not drawing on a tablet like I am. But I'm also trying to go with what I have, the equipment I have, and not buying extra equipment until I can make money on it. So anyway, the last thing I want to discuss really, and here is a diagram showing this thing again. The pathway starts at a rod and cone cell, who sends that information to a bipolar cell, who goes to the ganglion cell. From the ganglion cell, that axon of the ganglion cell makes the optic nerve. Optic nerve comes in. That information either crosses or doesn't, depending upon if that came from lateral visual field. It crosses over. If it came from medial visual field, it does not cross over. And the optic nerve goes to the optic chiasm that ultimately goes to the optic tract, which I left off of here. Need to actually have that on there. But what I will have it on there. I'm going to put that on there, but I'm not going to test on it this semester if I do, okay? Because your chapter review won't have that in there. And I apologize. Sometimes I catch little typos of things I intended. It's what you get when you type up 70-some, 80-some slides of material yourself. Nobody to help you edit. Okay, so we go to the lateral geniculate nucleus of thalamus to the occipital lobe to be understood. And that's what those projection fibers do. So you can actually get injury here and end up with like partial blindness of partial visual fields. So you have to kind of rework them backwards. It's interesting. By the way, there's a lot of, if you ever have to learn that in your studies on and on, is there's lots of pathway lesion charts out there that people have done, and for extra lesions like these. But basically, you can see what I've done here is I kind of try to do something a little bit like that as well. And there's lots of ways to draw it. I'm not the only one out there who draws it out. This is actually one of mine right here. A very popular video series that I've done for some other people, another company called Show Me. Anyway, I'm not trying to brag on myself, just making sure that you guys do learn that. It is a very common standard thing learned, but I like to draw it because it helps me to understand it. Now, the last thing I do want to talk about, it will not take us very long to do, is this is something you should have seen in AP1, but in case you didn't, let's go over it. Remember, there are two major types of general sense receptors, exteroceptors and interoceptors. Now, exteroceptors, they do things outside. But we've already done these. Vision, taste,
hearing. I don't know why taste got in there twice. So mine is as good as time as any to find those little issues and fix them. Smell equilibrium. So these are all things that we can see there as exteroceptors. These were specialized body condition things, external. Now, interoceptors are very important. These are internal conditions. Nociceptor should be pain. Do you want to feel pain? No, no, no. Nociceptor, pain receptor. Thermoceptors do temperature, mostly heat, but they do temperature. Cold is kind of the absence of heat. So or is the absence of heat. Mechanoceptors have mechanically gated ion channels. Now, these are things like tactile receptors, baroreceptors, and proprioceptors. Now, the type of mechanoceptor we saw was the hearing receptors, and the equilibrium receptors were types of mechanoceptors. But others out there are touch receptors, like the tactile receptors, lamellated corpuscles, tactile corpuscles. Baroreceptors are pressure receptors found in like the stomach and bladder, things like that. The baroreceptors, we're going to talk about a lot of those in cardio. The proprioceptors do body position. Those were like Golgi tendon organs, things like that to monitor tension. Those that are on the joint capsules, things like that to detect the position of the joints. Then there's chemoceptors. They do chemical concentrations. An important type of those is osmoreceptor, who checks the body fluid blood concentrations. Now, we will discuss throughout the different parts of the semester, we'll be discussing different types of it. Chemoceptors, osmoceptors, especially baroreceptors, things like that. We'll be discussing this throughout the whole semester. So these are big things, okay? So make sure you know what these basic things do. They are big deals. Now, at the end, I've given some basic diseases, just some things that I add to my notes. I always like to have some clinical little bits in there and nuggets, but you're not going to see those on any exams. So, guys, this does conclude my lecture on this, and I hope that you found it useful and helpful. Thank you so much. It will be on YouTube as well, so if you want to check it out there as well, please feel free. But for that, let's wrap it up. Thank you so much.